Okay, hi everyone. Today we're gonna be going over a lot of neuron stuff, which is really important for the SIBO. So just make sure you pay attention. We're gonna be going over neurons and how they signal, and then also certain nervous systems and like how signals are detected and stuff. Okay. So first is chapter 48, which is neuron synapses and signaling. So this is the basic like structure of a neuron, I guess. So neurons have a cell body and a really long axon and a bunch of dendrites. So the dendrites are where neurons will receive their signals, and axons will basically pass the signal on to the next neuron. So neurons basically transfer electrical signal information throughout the body to the brain and like back wherever. So the information is passed throughout the neuron, but then also transferred to the next neuron through synapses, which is right between the neurons, the dendrites, and the between the axons and the dendrites. So that's where the signal is transferred, and there are a lot of glia cells that also support the neuron, so they will provide a lot of nutrients and they will nourish cells, and they can also insulate certain axons. So glial cells include Schwann cells, oligodendrocytes, microglia, and also astrocytes, which we will discuss a little bit more in depth later. And like in nervous system, there is the central nervous system, which is the CNS, and the peripheral nervous system, which is the PNS. So the central nervous system consists of the brain and the spinal cord. That's where a lot of information integration goes on. And then the peripheral nervous system consists of like all the other nerves that connect the CNS with the rest of the body. And the PNS will also carry the sensory and motor information to and from the CNS. Okay. So there are three types of neurons in the body. There are sensory neurons, interneurons, and motor neurons. So sensory neurons basically detect an information from the environment and then pass that to the CNS for integration. Interneurons are interneurons are neurons that connect these two types of the sensory and motor neurons and also do some of the information processing. And motor neurons will transmit information out of the CNS to cause muscles to contract and other things to move. And so sensory neurons are also called afferent neurons. It starts with an A. And motor neurons are also called efferent neurons. Because they basically transfer information out of the CNS. So you can remember that because A is for like arrive at the brain, I guess, and then E is for exiting the brain. So yeah. Okay, and then the neurons function by they transmit their signals through ion pumps and ion channels. So neurons have a natural membrane potential, which is called it's called the resting potential. And so basically the neuron doesn't have a neutral charge. So inside of the neuron, it's a little bit more negative than the outside. And this is because there is a selectively permeable potassium channel that allows the outflow of potassium, but not the other ions. So potassium ion is a positive ion with a +1 charge. And so when the potassium flows out, then like your positive charge will leave the cell and then leave a negative intracellular environment. And there are also chloride ions, but they don't have a way of getting out, so the inside of the cell will be more negative. And then that also and also the sodium potassium channels will also contribute to that. So these channels will actively transport out three sodium ions for every two potassium ions that goes inside. So these the potassium ions will return back inside through the channel, but there are more sodium ions that leave than potassium ions. So if there's more positive charge leaving, then the inside of the cell will be relatively negative compared to the outside. And so the magnitude of the membrane voltage at equilibrium is called the equilibrium potential, and this is how you calculate it. It's 62 MV times the log of the ion concentration outside and the ion concentration inside. So this is an equation that's good to know because it was tested on a USIBO and they didn't provide, I think they provided the equation, but you have to remember the 62. And so it's just good for calculations. Yeah. Okay, so then these ion exchanges and ion channels will cause action potentials, which are the signals that are conducted by axons and that's passed on to neurons. So there in the neuron membrane there are some gated ion channels that will open or close in response to stimulus. So they're mostly usually closed, but then when there's a stimulus they'll open up. And so opening and closing will change the membrane permeability to certain ions, which causes a really rapid change in the membrane potential. And these are called voltage gated ion channels. Sorry, there's no text, but there are voltage gated ion channels that will basically respond to a shift in the voltage across the membrane, or they're regulated by chemical ligands, and then that will cause them to open up. So this is the graph of an action potential, and this is basically what happens when your neurons like transmitting a signal. So at resting potential, the membrane potential is around -70, but then so then depolarization happens, and depolarization is when your gated sodium channels will open up in response to a stimulus, and then that will allow more flow of um sodium into the cell. So remember how sodium is being pumped out of the cell? Well, these channels will allow the sodium to come back into the cell because of the gradient. And then since sodium is positive, that will make the membrane potential more positive. And then that's called depolarization because your membrane was very polar and now you're depolarizing it with sodium ions. So then that will continue until you hit the threshold. And once you hit the threshold, the action potential will happen. You have a rising phase and then your falling phase. And then this coming down part is called hyperpolarization. So during this time, gated potassium channels will open to allow more flow of potassium out of the cell. So then that will make the cell more negative, and that's why it's called hyperpolarization. And then eventually eventually the sodium channels will be deactivated, but the potassium channels will remain open, and that is why like at one point the membrane potential falls under the resting potential, and that's called undershoot. And then everything like resets and stuff. Okay. Um and oh, also it's good to know that an action potential is an all or a non-response. So if so once your membrane potential like hits the threshold, the action potential is going to happen. It's not like it's going to happen halfway or anything. It's going to happen. And so there's no like level of action potential you can have. Okay, and then so eventually the membrane potential will get restored, but so immediately after an action potential, like a second one cannot be fired, and this is because of the refractory period. So during hyperpolarization, the sodium channels will get deactivated, and that allows for the potassium to like restore the membrane potential. But then the sodium channels will remain deactivated for a little bit, and then that's called refractory period. And so the charges will not move back through the membrane, and then you can't have a second action potential immediately following the first. Okay, so this is the mechanism of propagation of the action potential. So first the sodium comes in, and then these charges inside and outside of the cell will interact. So that the positive and negative charge will interact, and then the positive charge will start to move along the cytosol. And then like behind the action potential, the potassium is flowing outwards during hyperpolarization. And it is also undergoing a refractory period. So even though there is a negative charge here, the positive charge from the sodium here, even if it moves to the left, it's not going to cause another action potential because the sodium channels are inactivated for that period of time. Okay, and action potentials are aided by myelin sheath, which is basically a layer of electrical insulation around the axon. So the myelin sheath is made by Schwann cells or oligodendrocytes, and they will act like an insulator, which doesn't allow for the flow of electricity. So so myelin sheath will speed up the propagation of an action potential. And this is because they have certain nodes of Ranvier, which are areas without the myelin sheath. And so basically the action potential can only happen at these nodes of Ranvier because these other parts are all insulated. So that will essentially let the charges jump around the nodes and like go way, way faster than if you had a single axon and had the charges go through the entire length of the axon. Okay. And then this jumping process is called saltatory conduction, and salta is basically jump. It means jump. Okay, yeah, this is just the mechanism. So there will be the charge difference, but then it's gonna only happen at the nodes of Ranvier, and it's gonna move way faster. Okay, so and then neurons will communicate with other neurons or other cells at synapses. So most synapses are chemical synapses where chemical neurotransmitters are released through synaptic vesicles and will bind to ligand-gated ion channels at the other side. So these neurotransmitters are released when an action potential reaches the end of a presynaptic neuron, and that will cause a calcium influx from the outside of the cell, and then that will then release neurotransmitters in the synaptic vesicles. And when these neurotransmitters bind to the ligand-gated ion channels, they will produce an excitatory or inhibitory postsynaptic potential. So an excitatory postsynaptic potential will cause the postsynaptic neuron to like be more activated, while inhibitory one will cause it to be less activated. So here are the major, like the most important neurotransmitters. The first one is acetylcholine. It's very important because it allows for motor function, and it's very commonly um it's it's very present, I guess. Okay. And then too little of it will can lead to Alzheimer's, which is really bad. Next are the amino acids. First is glutamate, which is excitatory. It's responsible for long-term memory. And if you have too much of it, you get like migraines and seizures and stuff. And the next one is GABA aminobutyric acid, also known as gamma aminobutyric acid, also known as GABA. And that's responsible for reducing anxiety and also responsible for relaxation. And glycine is important, but it's not as important. And then the next ones are norepinephrine, dopamine, and serotonin. So norepinephrine is responsible for speeding up the heart rate and regulating blood pressure when you're like under stressful situations, for example. Dopamine is responsible for the short-term like reward feeling, and serotonin is responsible for like a long-term like good stable mood. And then nitric oxide, which is also really important, it's responsible for vasodilation. Okay, next is nervous systems. Okay, so nervous systems will consist of neurons and their supporting cells, which include the glia cells. Okay, so many animals will have a central nervous system and peripheral nervous system that will carry the information out. And these are like the very the various types of nervous systems that organisms can have. And the more complex animals have ganglia, which are segments of neurons that are relay points in information transfer. So like for example, if you look at the salamander, it has little like sensory ganglia, and also the squid does too. But like the less complex animals, they don't have that. Okay. So, nervous systems have evolved a lot. So, initially there were nerve nets where neurons were scattered randomly. So, like in the Hydra, which is a cnidarian, it had a very randomly scattered nerve net. And then that evolved into nerves, which are bundles of neurons. So, as you can see in the sea star, it has like a nerve ring and radial nerves. And then cephalization evolved, which is the clustering of sensory and processing neurons at the head. Um, and then the central nervous system and peripheral nervous system evolved. Yeah. So like as you can see here and also in the salamander, there are clusters of nerves at the head. Okay. So the vertebrate nervous system has evolved from that, and the vertebrate nervous system has glia cells which will support neurons. So like I said before, Schwann cells are make up the myelin sheath of the peripheral nervous system, and the microglia are the immune cells for the central nervous system. Oligodendrocytes are the make up the myelin sheath of the central nervous system, and astrocytes will promote blood flow, transfer nutrients, act as stem cells, and it has a lot of different functions in the central nervous system. And also ependymal cells will help circulate cerebrospinal fluid in the brain. No. And also interactions between astrocytes and microglia can promote neuroinflammation, which can or cannot be good depending on the context. Um and Okay, so basically in the brain there are gray and white matter. So the gray matter is all the neuron cell bodies and the white matter is mostly axons. And the spinal cord, it's white on the outside and gray on the inside. Um and for the brain, for the general nervous system, um so the spinal cord is called the central canal, and the brain has a lot of ventricles, and they're filled with cerebrospinal fluid, which helps like cushion the brain. Yeah. Oh, and in the central nervous system, the brain will process information regarding like emotions, complex thoughts, learning, and information processing, and the spinal cord will bring information to and away from the brain, and it can also generate reflexes, which is when the spinal cord will act independently of the brain and produce automatic responses. So for example, to like pull your hand away from a really bad stimulus. Okay. Okay, and the peripheral nervous system is everything else that will bring information to and from the brain and from the central nervous system. So there are two divisions, the motor and autonomic nervous system. So the motor nervous system will cause voluntary movement responses. So like when you want to walk, that's what's controlling the walking. And then the autonomic nervous system will control involuntary responses of smooth muscle, glands, and like cardiac muscle. So then the autonomic nervous system will divide into the sympathetic division and the parasympathetic division, and also the enteric nervous system. So the sympathetic division is responsible for arousal, like when you're nervous, maybe like your heart will accelerate, um your digestive activity is going to be inhibited, and also like glucose will be released from the liver and everything. And then the parasympathetic division is responsible for relaxation, where the pupil will constrict, the heart rate will slow, and digestive activity will be restored. And then also, the enteric nervous system has direct control over the digestive tract, the pancreas and the gallbladder. And the sympathetic nerves will exit the spinal cord in like the midsection from the thoracic to the lumbar sections, while a lot of the parasympathetic nerves will exit from the cervical up in the higher sections, and also the lower sections. Okay, so the vertebrate brain has three sections: the forebrain, the midbrain, and the hindbrain. So the forebrain is responsible for the processing of complex thoughts, emotions, and senses. So like if you were thinking of what you're gonna eat tomorrow, that happens in the forebrain. The midbrain will kind of route the information to the forebrain, and the hindbrain is responsible for really vital involuntary actions like the circulation, heart rate, breathing, all involuntary breathing, all of that. So generally for mammals and birds, the forebrain is larger than other parts, and they and creatures with larger forebrains are generally better at complex thinking and processing. And so as you can see here, humans have a huge forebrain, and that means they're really, really good at processing information like we are. Okay. So here is the vertebrate brain continued. So the big part of the brain is called the cerebrum, and it's divided into four lobes. The very outer thin layer of the brain, so the entire thing's called the cerebrum. The outer layer, it's kind of like the shell-ish. The outer layer is called the cerebral cortex, and there are two hemispheres in the brain. They are connected by cells called the corpus callosum, and each hemisphere will receive information from the other side of the body. So, for example, like when I like touch something with my right hand, my left hemisphere is receiving information from it. And in general, the right hemisphere is more responsible for creative activities, and the left is more responsible for logic-based activities. And this difference in function is called lateralization. And the four lobes of the cerebrum are the frontal lobe, temporal lobe, parietal, and occipital lobes. So the frontal lobe, which is the big blue thing here, it's responsible for decision making, planning, and complex thinking. It also has Broca's area, which is responsible for forming speech. The temporal lobe, which is at your temples, is responsible for hearing because it's really close to the ears. And it contains Wernicke's area, which is important for comprehending language. And then the parietal lobe is like right behind the frontal lobe and like the back towards the top. It's mostly responsible for integrating sensory information, and it contains the somatosensory cortex, which will like route your sense of touch and everything. Um And also in the back of the cerebrum is the occipital lobe, which contains the visual cortex to integrate visual information. So then the cerebellum, which is below that, it's the green part, it controls more motor coordination. The blue part is the brainstem. It contains the medulla oblongata, and it controls vital functions such as breathing and heart rate. And then the midbrain has a bunch of little things. I don't know if I have. Oh, yes, I do. Okay, the midbrain is the middle. It has a bunch of little parts, but they're all very important. So it includes the hypothalamus, which controls the pituitary glands, which then controls hormones, and it basically regulates temperature, hunger, your sex drive, and also emotions. Um, it also contains the thalamus, which helps route all the senses except for smell. The smell is routed from another part of your brain. It also contains the reticular formation, which controls arousal from sleep, contains the pons, which controls other vital functions and motor coordination, contains the suprachiasmatic nucleus, which controls the circadian rhythm, which is also stimulated by the presence of light. It has the amygdala, which controls emotion and like anger. The hippocampus, which helps with memory, And it's also good to note that the hippocampus, amygdala, hypothalamus, and thalamus are called the limbic system. Okay. And the brain can be scanned in many ways. So the first scan that I've shown here is the electroencephalogram, which is the EEG that basically detects brain waves and brain activity. This is position emission tomography, the PET or the PT scan. That's when you like a person intakes radioactive glucose and then the glucose will circulate in the brain and that will show where the brain is most active. Oh, I think this was an MRI. It's where you have magnetic waves and then that shows the structure of the brain. CT scan, computer tomography is doing a similar thing to MRI but using X-rays, and functional MRIs can show like functions of brains rather than just the structure. Okay. Um, so changes in synaptic activity will help with memory and learning. Neurons have a property called neuronoplasticity, which is the ability of nervous systems to remodel and rebuild itself after damage. So, certain parts of the brain can replace other parts' functions. So, when you have like a little child and they must have part of their brain removed for whatever reason, like a non-vital part of the brain, like for example, like in the frontal lobe, maybe other parts of the brain can kind of take over that and like do functions of part of the frontal lobe properly without like yeah, they can do the functions properly. And there are short-term and long-term memory. So short-term memory is usually like a couple of seconds after the event happens, and if it's not important, we'll release that information, never recall it again. But if it's important to us, we will encode it into long-term memory. And memory is stored in the cerebral cortex, but the hippocampus will act kind of like a link to retrieve the memories. And neurons can also undergo a long-term potentiation, where increased stimulation of a synapse will create stronger and better synaptic activity. So this is shown here. So there is a lot of stimulation a bit in the synapse between N1 and in the cell body, and less between N2 and the cell body. So like eventually N1 ends up establishing more synapses, more synaptic clefts with the cell body, and N2 removes its synapse. There are also some brain diseases. Parkinson's is due to a lack of dopamine because of the death of dopamine releasing neurons. Schizophrenia is a distorted perception of reality. Alzheimer's is a mental deterioration which is associated with aging. So a lot of old people might get Alzheimer's. Addiction, which is when you have this really addictive substance that can mimic neurotransmitters, make you feel really good, and then your neurons will create a lot more a lot more receptors for that substance, and then you will constantly need more and more of it, but once you don't have it, you'll go through withdrawal and it's really, really painful. And also depression, which is where otherwise enjoyable activities provide no pleasure or joy. And a sub part of that is bipolar disorder, which a symptom of it is extreme mood swings. Okay. Okay, next up is sensory and motor neurons. So, sensory reception is where a stimulus is detected in a receptor cell, and the most common response to this is opening or closing of channels to change the receptor potential. And the conversion of sensory reception to receptor potential is called sensory transduction. And sensory transduction will happen through action potentials. And the size of the receptor potential will increase with the stimulus, because there will be more frequent action potentials or more neurotransmitter released, like per transduction. And then the sensory information will be processed in the brain. Okay, so this is what I was just talking about before. So when there is gentle pressure on the sensory receptor, there will be lower frequencies of action potentials, but then when there's more pressure, there will be more stimulus, and then that will produce a higher frequency of action potentials per receptor, and that will be eventually integrated and interpreted into the brain. There are a lot of sensory receptors, and they will tell the brain stuff about the environment. So mechanoreceptors will respond to touch, pressure, sound, motion, and stretch, and there are a lot of them in the skin. Chemoreceptors will transmit information about solute concentration, such as glucose, salt, oxygen, and carbon dioxide. A lot of smaller animals will have those, such as like moths. I think they have chemoreceptors for, I think, glucose. I'm not really sure, but they have something like that. Thermoreceptors will detect heat or cold, and nociceptors are also they're also called pain receptors. They will detect pain and trigger defensive reactions. And lastly, electromagnetic receptors will detect energy like light, electricity, and magnetism. And certain birds will have this, and also other animals. They will help animals navigate by detecting the Earth's magnetic field and telling them basically where to go. Okay, so this is sensing of gravity and sound in invertebrates. So invertebrates will have statuses, which are kind of like little bubbles. So the outside layer are a layer of cells with cilia, like little hairs. And then inside of that little statocyst, there will be statoliths, which are kind of like little um pieces of, I don't know how to say it, like little particles. And then when the animal repositions or moves around, the satellites will also move around with the help of gravity. And then that will move the cilia, um, which will affect the ciliated receptor cells, and then they will send information to the sensory nerve fibers, and that will go to their nervous system. And then so this helps the animals detect position. And oh, so this is and the human ear will also do that. Well, it will detect position of the human, but also will help detect sound. So the outer ear is called the pinna. The very outside part is called the pinna, and the outer ear will consist of the pinna, auditory canal, and the tympanic membrane. So the auditory canal is like the path to the tympanic membrane, which is the eardrum. The eardrum will separate the outer ear from the inner ear and also collect vibrations. And the middle ear will consist of three bones: the malleus, which is the hammer; the incus, which is the anvil; and the stapes, which is called which is also the stirrup. And it also consists of the eustachian tube. And the three bones, the malleus, incus, and stape, will transmit the vibrations from the tympanic membrane to the oval window. And the eustachian tube will connect to the pharynx, and it will equalize air pressure. So that's why when you like that's why when there's a air pressure difference, you can yawn. That will like open up the eustachian tube and help regulate air pressure. And in the inner ear, there are many fluid-filled chambers. So the oval window will connect to the cochlea. Will connect to the cochlea here. And the cochlea will have an upper vestibular canal, which is separated from the lower tympanic canal by the cochlear duct, and the organ of Corti, which is inside the cochlea. It contains mechanoreceptors of the ear, which will help with balance, with orientation, and all the stuff like that. Oh, and the semicircular canals will contribute to equilibrium. Okay, here is how hearing works. So first, the sound is amplified through the liquid in cochlea, and it can create pressure waves. So that happens inside of this part. And the pressure waves will, so the cochlea will have a lot of hair cells, and then the pressure waves will deflect the hair on the hair cells. And bending the hair in one direction will depolarize the cell, emitting in the other direction will hyperpolarize the cell. And so these pressure waves are dampened when the waves will travel across the apex of the cochlea, hit the round window, which is on this side. And the movement of the hair cells will capture information about the volume, which is the amplitude of the waves, and also the pitch, which is the frequency of the waves. So louder sounds are correlated with more intense pressure waves, and higher pitch is so higher pitch is detected when each section of the basilar membrane vibrate differently based on different frequencies. Okay. And equilibrium is detected by the semicircular canals. The utricle and the saccule will detect the position and motion with respect to gravity, and they contain hair cells which are embedded into a gelatinous material that contain otoliths, which are basically ear stones, I guess, that shift the position and brush against the hair cells. So this is a little similar to the stelloliths that we saw earlier. And also semicircular canals have hair cells that project into the cupula, which is like a jelly-like material. And when the head moves, the perilymph, which is the stuff inside the cupula, the fluid inside the cupula, it's gonna move and bend the hairs and transmit information about the body's movement. Okay. Wait, I'm gonna stop here for a bit if there's okay. Okay, there's no questions. Hold on, I lost my page. Okay. Okay, and in other vertebrates, I guess this is just equilibrium. The this is good to know because it was also on a question in the USIBO. Fish don't have ears, but their vibrations are the vibrations of the water, and their orientation is detected through the skeleton. And they also have a lateral line system that will detect low frequency waves and anything that changes in the environment. So they have hairs in the lateral line that are also covered by a cupula. And when the cupula is bent by water, the hair cells are also bent, and then that will relay motion change, velocity change, or like water flowing over the body in general. Okay, so next we'll go on to the light detecting organs and the eyes and everything like that. So the general concept is photoreceptors will contain light absorbing pigments, and then that will convey to the brain, to the nerves, like what's going on with light, with frequency of light and everything. So the most simple are eye spots, which are called ocellus. Planarians have that, and these help planarians move away from light. So this is because any light striking the back of the eye spot is blocked, but then striking the front is transmitted. So that will help them tell where they are, like, oriented to light. The next most complex is the compound eye. These compound eyes will have several thousand ommatidia, which are facets of the eye. They're like mini eyes, essentially. Each one will capture a little bit of the visual field. And so if you have a lot of them, you can see like all around you. And each will have a cornea, a crystalline cone lens, and a photoreceptor, so each will act like a mini eye. And humans have the single lens eye, which we're going to talk the most about. So the single lens eye is like a camera because the light will enter through the pupil, which is adjusted by the iris. And the iris is like the shutters that will adjust how much light goes in. The light will directly hit the back of the back of the eye, which is called the retina. And the cornea on the very outside will protect the lens and the rest of the eye, and it's cushioned by the aqueous humor. And I guess just good to know, blockage of the ducts that drain aqueous humor is really, really bad, and it can cause glaucoma, which is vision loss due to the increased pressure on the optic nerve. and inside the eye is filled with a jelly-like substance called the vitreous humor. And anyways, more about the retina. The light will strike the retina, and that light will reach rods and cones. So rods will detect features of objects such as like lines, shapes, whatever. And they're more sensitive to the presence of light. And so they're mostly responsible for your vision in the dark. While cones will detect color, they detect the wavelength frequency of the light. So this is also why you can't really see color at night because your cones um are like less sensitive to low light conditions. And when the light strikes the rods and cones, the signal will pass to bipolar cells, which will then pass to ganglion cells, which then pass the signal to the optic nerve, and then that moves to the brain. Um, okay. And er, on the back of the retina, there is an area called the fovea. That is where you can see the clearest. Like that's where most of the light will focus onto the fovea. But there's also a blind spot in the eye, which is right when the optic nerve like starts to form because as you can see here, like at this part of the retina, there are no rods and cones, so in that part you can't really see. Yeah. Okay. So more about single lens eyes. The rods will detect presence of light, cones will detect the frequency of light and color. And they contain discs with visual pigments. Um These pigments are retinol and opsin, which are bound together, and that's called rhodopsin together. And the presence of light will shift the cis-rhodopsin to a trans-rhodopsin. So the cis isomer is bent, the trans is straight, and that will activate a G protein, and that will like allow for light to relay. And also, retinol is a derivative of vitamin A, so getting vitamin A is good for your eyes. Eat some carrots or something. Okay, so here's the sensory transduction in the eye. So the light will hit the photoreceptor. That's gonna change the cis-rhodopsin to trans-rhodopsin. That's going to activate a G protein. And then that G protein will activate phosphodiesterase, which will then create cyclic GMP that binds to the sodium channels and keeps them open for depolarization. And then once the cyclic GMP is no longer in use, it will be hydrolyzed, the sodium channels will close, the cell will undergo hyperpolarization, and the rhodopsin will be changed back into the cis isomer form. Okay. And this is just a comparison of light and dark. So in the dark, the rhodopsin is inactive, it's in its cis form. And in the light, the rhodopsin is active, it's in its trans form. And in the light, the sodium channels are closed. In the dark, the sodium channels are open and they are depolarized, hyperpolarized. And in the dark, glutamate is released. And in the light, there's no glutamate, and the bipolar cells their activity will depend on the photoreceptors. Okay, and the optic nerves will converge in the middle, and they will meet at the optic chiasm in the brain and cross to the opposite hemisphere. And they will eventually reach the primary visual cortex in the back of the occipital lobe. Okay, and here's the visual field. So color vision is controlled by red, green, and blue visual pigments in the cones, which results to binding to different opsin. Oh, this is because retinol can bind to different types of opsins, which creates different pigment receptors. The stimulation of these receptors in combination will create perception of different colors. And so like for example, if you have stimulation of red and blue, they can create a purple illusion, but if you have more red, it becomes more and more magenta and pink. And color blindness will result from mutations in pigment genes. And the most common color blindness are green, red, and blue, yellow. And color blindness is a lot more prominent in males than females because color blindness is an X-linked mutation. So males will be affected by a single copy on the X chromosome, but females will only be affected if both chromosomes are affected. So, like I said before, the vision is the clearest at the fovea, at the point in the back of the retina. And so all light should ideally be focused there by changing the shape of the lens. And the fovea also contains a large proportion of cones to rods. So in the daylight, we will see the clearest by looking directly at the point of focus. However, in the peripheral vision, there are a lot more rods than cones. So like around the fovea, and like in the points where we don't focus, there are a lot more rods than cones. So this is also why in the dark we see better. Like we see more clearer by looking slightly away from the object. So yeah, that's pretty cool to know. Okay, next is the sense of taste. The taste is also called gustation. It's essentially where tastins, that's like the little particles, they will come into contact with taste pores on taste buds and bind to specific sensory receptor cells that will transmit information about the taste of the tastin. And the tastins are specific to the taste and can be a variety of different molecules. So for example, for a salty taste, you can just have like table salt. For sweetness, you can just have glucose, that can be the tastant. And acids can help transmit acids can be the tastant for the for the sour taste. And olfaction is smell, so this is kind of similar to taste. The receptor cells are neurons themselves instead of a specific sensory receptor. So the cilia of the neurons will extend into the mucus to detect the presence of odorants. And these odorants can also be a variety of different molecules. And the binding of the odorant will activate GPCR to produce cyclic AMP, and that will open ion channels for depolarization. So the senses of smell and taste are very interconnected and also will interact. So like when you eat with a stuffy nose, you can't really taste what you're eating. That's because of the interaction between smell and taste. Oh, and also just interesting to know, the olfactory bulb of the brain is located very closely to the hippocampus. So a lot of memories will be triggered by certain smells. Okay, next is skeletal muscle, which is kind of complicated. So skeletal muscles are made of muscle fibers, and these muscle fibers are made of will have thin filaments, which are actin, and thick filaments, which are myosin. And those filaments will help with the contraction of the muscle. So each muscle fiber is a single cell with multiple nuclei. And myofibrils, which are arrangements of thin and thick filaments, will surround the nuclei and are composed of repeating sarcomeres, which are just basically like the line patterns. And because of this, they're also called striated muscle because of the striation lines. So in a resting muscle, the actin and myosin filaments are slightly overlapped. The myosin is the thick filaments are arranged in the middle, and the thin filaments are arranged on the outside. So the actin filaments are anchored at the Z line, the myosin filaments are anchored at the M line, and then the middle part is the H zone, which is the part where there is only myosin, no actin. And oops, my bad. Okay. So here's the sliding filament model. So in this sliding filament model, when muscles contract, the actin and myosin will just kind of slide against each other, and that's what causes the muscle to contract. So each myosin molecule has a tail and a globular head. globular head here will attach to ATP and also actin to push it along to essentially slide the filament along for contraction. And ATP will power the movement of myosin. And the tail and the globular head are called the cross bridge together. And an energized cross bridge will bind to the actin, kind of push it backward, and then release in its low energy form. And the myosin cross bridges will move along the actin basically. And the cross bridges on the two ends are oriented in opposite directions so it can push the thin filaments towards each other. This is a huge use of ATP. So muscles use aerobic respiration to produce ATP during light activity, and then we'll use glycogen and lactic acid fermentation when oxygen runs low. And that lactic acid will contribute to a sore feeling after muscle use. And different muscles have different ways of producing energy. So some muscles will be oxidative muscles and some will be glycolytic muscles. Okay, and muscles will not always be contracting, obviously, and this is because of calcium and other regulatory proteins. So, usually there is a protein complex called tropomyosin, and that is wrapped around the actin to prevent the myosin from binding to it, and that is also held in place by troponin. And this troponin is released when calcium binds to it. So when calcium comes in, the muscle can start contracting again. And this calcium is triggered by neuron action potentials. So for like if you have a neurotransmitter, for example, acetylcholine come in, um the action potential will what's it called diffuse down the transverse tubule or the T tubule that will go along the sarcoplasmic reticulum, like a form of the endoplasmic reticulum, but in muscle cells. That will cause the SR, the sarcoplasmic reticulum, to release calcium, and then the calcium will then bind to the troponin, which will release the tropomyosin, and then allow for the myosin to bind to the actin, and then cause the muscle to contract. And a muscles can have graded contractions. It's not like neurons. There can be several levels of muscle contractions. And that all depends on the number of fibers that are stimulated and the frequency of stimulation. So when you have one action potential that hits the neuron, it hits the muscle, it produces a twitch, and multiple action potentials can undergo summation. And once you have high enough summation, that leads the muscle to go to tetanus, which is one smooth muscle contraction. Oh, and also a motor unit is one motor neuron and all the muscle fibers that it controls. Okay, there are three different types of muscle fibers. The first type of muscle fiber is skeletal muscle. That is the muscle that produces a lot of your voluntary activity. There are oxidative and glycolytic muscle fibers. The oxidative muscle fibers use mostly oxidative respiration, anaerobic respiration to produce the ATP supply. So it's going to have a lot of myoglobin to store oxygen. It's going to have a really large blood supply to bring the oxygen and bring away wastes, and also mitochondria for the oxidative of phosphorylation. And glycolytic don't rely on that as much. They can undergo glycolysis to produce its ATP, so they don't need as much of myoglobin, mitochondria, or blood supply. There are also fast twitch and slow twitch fibers. So fast twitch fibers twitch really quickly, while slow twitch fibers don't do that as much. There are three main combination types. There are slow oxidative muscles, fast oxidative muscles, and fast glycolytic muscles. And slow glycolytic muscles don't really exist. Yeah. And in general, glycolytic muscles are a little thicker and can hold more tension than the oxidative muscles. So cardiac muscles are found in the heart. The heart has certain cells that will act as pacemaker cells to initiate the contraction instead of neurons. And the action potentials in the heart will last longer, but also will have a longer refractory period to prevent tetanus. So in the heart, you don't want a constant heart contraction because that's just really bad for the heart. Yeah. And then smooth muscle is in a lot of your blood vessels. It's also in a lot of your organs, like your intestinal tract. And they are stimulated by the autonomic nervous system, and they have scattered myosin and actin that are arranged nicely, and they don't have a troponin and troponin system or a T-tubule, so the calcium will enter through the plasma membrane and bind to calmodium, which initiates the myosin head binding. Okay, and I have a couple of slides left. So muscles have a flexion and extension muscles. Extension muscles will like pull your limb longer. And the flexion muscle will like pull the limb like more contract into their more contracted form. Okay, and here's just a really quick thing about skeletal systems. There are a lot of different skeletal systems. The hydrostatic skeleton will basically hold all the fluids in the body, and the muscles will change shape of the filled compartments, and organisms can move by peristalsis, like in a worm or in a hydra. And the exoskeleton is a hard covering deposited on an animal's body to protect the soft insides. A lot of bugs have that, and it consists of chitin and calcium salts. And humans have an endoskeleton, which is a skeletal system inside of soft tissues. Vertebrates have cartilage and bones are made by osteoclasts and destroyed by osteoblasts. Okay, and this is just the joints of the endoskeleton. So the ball and socket joint provides rotational motion. The hinge joint provides like bending motion, and the pivot joint is rotation, but like sideways, like in your wrist. Okay. Okay, we are good. We are done. You gotta go. You gotta go.